Hi, everyone. I'm delighted to welcome Dr. Sarah Nevitt from the University of Liverpool to our Vivli webinar. Sarah has a background in math and statistics with a bachelor's degree from the University of Liverpool and a PhD in biostatistics also from the University of Liverpool. She's currently in the Department of Biostatistics in Liverpool, and she supports Cochrane authors in the Cochrane Epilepsy Group, and she's co-authored 8 IPD reviews in epilepsy. So we're really happy to have her. We think she's the ideal person to walk us through some of the an introduction to IPD analysis, meta-analysis using the Vivli platform. And thank you very much for being here, Sarah. We're really looking forward to your talk today.
Well, thank you very much for the introduction, Rebecca, and to everyone from Vivli for inviting me to give this webinar today to talk a bit about the research that I've done in this particular technique that I use and how data that is available on the Vivli platform fits into this sort of analysis. So what I'll be talking about today in this webinar is individual participant data, or IPD to shorten it, meta-analysis. So I'll firstly start with just a brief introduction to the use of IPD in a secondary analysis such as meta-analysis, what this means for analysis techniques, when you would use this sort of technique and why you would want to use it. And I'll be talking about some of the work that I've done from the Cochrane Epilepsy IPD review group, and also some of the practical aspects of conducting that sort of work rather than any great detail about the actual methodology behind it. I'll also be talking about something which is very key to some of the research that I've been doing in some of my PhD work behind the era of data transparency that we're now in for the last 5-10 years, with great pushes towards sharing data throughout the entire research community, and what this means for IPD meta-analysis. And then finally, I've got a few slides just to give a few ideas and tips about what to do in the probably quite likely scenario of not all IPD being available for the analysis that you want to do, and what you can do about that. So I'll get started. So firstly, starting with a couple of definitions. I'm going to define a primary analysis as being the original analysis of clinical data. And that's usually the analysis that is done for the purpose that that data was collected. So data being collected for a randomized control trial, or maybe an observational study, a survey, and so on. The first analysis done of that data would be the primary analysis. Whereas a secondary analysis is any subsequent analysis of that clinical trial data, so a second, third, fourth, and so on analysis, which may be done for the same purpose as that data was collected. So in other words, a reanalysis, maybe by the same team, maybe to correct something that wasn't quite right, or maybe by a different team to verify the results. Or an analysis could be done for a completely different and novel purpose, compared to why the original data was collected. To give a few examples, certainly not an exhaustive list, but just a few different techniques which would be classed as secondary analysis: systematic reviews and meta-analysis of secondary analyses, as are prognostic models, development and validation of those sorts of statistical models, any maybe detailed evaluation of a drug profile, such as long-term consideration of adverse events across different studies, would be a secondary analysis. And also, primary data may be used to inform future work. So the primary data may be used to determine the new design or the new sample size of a new study, and this would also be classed as a secondary analysis of that data. So a few considerations when thinking about doing a secondary analysis, a research question which involves a secondary analysis using IPD from primary studies, and what to include within the formulation of that research question. Firstly, it's very important to articulate why that secondary analysis is required, whether it is a reanalysis or a different analysis. Is it because the first analysis didn't answer the question, or perhaps the question should have been different, or the question should have been answered in a different way? Maybe different outcomes, patient groups, different methods, or additional methods could have been used in that question. It's important to provide clear scientific rationale behind that secondary analysis question to support why different outcomes, different patient subgroups, different methods and so on should be used in this secondary analysis, and specifically how the IPD is going to be used. Will it be a reanalysis of every patient, or perhaps a subset of patients, or will this data be used maybe to help design a new trial? Additional considerations for secondary analysis research questions, such as systematic reviews and meta-analysis, we may be looking at IPD from multiple primary studies and maybe combining that data across multiple studies. Thinking about the studies that will be included within this sort of research, it's important to outline clearly the inclusion criteria in relation to this analysis question. And a good framework to do this is the PICO framework. So P being patient, population or problem, I being intervention, C comparator, and O objectives or outcome. It should be possible in the research proposal, the formulation of the research question, to clearly and specifically outline what each of these PICO criteria is. And also when selecting studies which may or may not meet the inclusion criteria to include in the analysis, and also the IPD may be requested for, think about, will this be systematic work where a systematic search of databases is done and perhaps all relevant evidence for the PICO is included, or perhaps maybe there may be some restrictions on which studies will be included in the analysis work. So maybe only the most recent studies will be included, maybe only studies from certain areas of the world would be included, or if studies aren't available with IPD, they may be excluded. So depending on whether the research question, well, regardless of whether the research question uses one study or multiple studies, it's helpful to have a clear research proposal or structured protocol outlining every possible element of that research question before requesting any IPD. And just a few words on Cochrane Collaboration, the organisation that I've supported for the last 7 years. I've worked with many of the clinical review groups. There are 55 different clinical areas under Cochrane, and the main group I've worked with is epilepsy. So it's an organisation of lots of different people, so researchers, methodologists, health professionals and patients, thousands of people all over the world. Also a lot of methodological work going on with 17 review groups. And the Cochrane collaboration follows a structured and systematic approach to synthesizing data in order to provide the highest quality evidence on a clinical question to be able to aid decision making. So what is meta-analysis? Getting a bit more specific now on the techniques that I'm actually using in my work. So meta-analysis is a statistical technique for combining sources of quantitative or numerical evidence. And I'll be talking about two different types of meta-analysis today in the work that I've done. Firstly, pairwise meta-analysis. This is the traditional meta-analysis, which is in most Cochrane reviews. So this is the evaluation of two different things, so two interventions, maybe intervention versus control, drug versus placebo, which have been compared head-to-head in 2 or more clinical studies, and that data can be combined across the studies to get an idea of how those interventions or the interventions and control compare to each other. And then also network meta-analysis, which is an extension of the traditional pairwise meta-analysis to allow more than 2 interventions or multiple interventions, multiple controls, and to be included and combined where there's more than 2 studies looking at those interventions. The benefit of network meta-analysis is this also allows indirect evaluation of interventions which have not been compared head-to-head in any clinical studies. So actually this technique allows us by statistical methods to have an estimate of the relative effectiveness of all interventions in the network without actually having to conduct new studies. So this is very helpful for decision making. And throughout the methodological slides that I am describing in this webinar, I've given references throughout for further reading on these methods. So please do consult these good references if you're interested. So meta-analyses have traditionally been performed with aggregate data. So what do I mean by aggregate data? So summary statistics such as mean differences, event counts, odds ratios, risk ratios, hazard ratios, and so on, that you will find in the published literature. So journal articles, conference abstracts on trial registries such as clinicaltrials.gov. So for example, you look in a journal article, you find a table of results that looks like this one. You could extract from here the number of events. You might also want to extract some of the relative effects of how the drugs compare to each other. So here we've got some hazard ratios. And also more recently, there's been recommendations around how useful unpublished documentation is, such as protocols, statistical analysis plans, and clinical study reports are, for providing additional information to be able to include within meta-analysis. So the alternative to doing an aggregate data approach is an individual participant data, patient data, IPD approach. So in this approach, the original participant level data, line by line, is requested and reanalyzed. So I've given a simple example here. This is just a table of data that I've completely made up, which has got a patient ID number, the treatment that they're on, some demographic information such as age and gender, and also some information here relating to an outcome. So here we're looking at how long someone was taking a drug for before they withdrew from it. And an IPD approach is widely regarded to be the gold standard approach to meta-analysis, as there are many advantages to this approach. So what are these advantages? Essentially, using an IPD approach allows a more flexible and complex analysis approach. In very simple terms, it allows a researcher to do whatever they want to do with the data, rather than having to rely on what has previously been done and previously been published about that study, about that data. So some of the techniques which are possible with IPD, outcomes can be standardized if there's any differences across studies, or redefined if there is an interest in looking at an outcome a different way. It may be possible to reinstate participants in a study who may have been excluded, for example, due to protocol violations. In theory, an IPD approach will reduce biases related to meta-analysis, such as publication and reporting biases, as unpublished information is requested. And also in theory, an IPD approach may reduce what is known as ecological bias, which can be an issue in aggregate data meta-analysis. In other words, the average effect across a group may not necessarily reflect the individual response to a treatment. So to give an example, the effect of a drug on a group of people with an average age of 50 might not necessarily represent the effect of that drug on one person of the age of 50. That's what IPD could be used for, to model more patient-specific information. So IPD allows detailed checks of any assumptions that have been made within statistical analysis, and it also allows more complex analyses to be done. So in investigating patient level information, so perhaps the effect of different covariates on different treatment effects, maybe modelling heterogeneity, so variability within and between studies, and also modelling complex data types such as prognostic data or diagnostic data within synthesis. IPD approach would be considered gold standard for this. So in addition to all of these advantages, actually an IPD approach may be the only option where the summary level aggregate data that is needed, so the summary statistics needed to go into a meta-analysis, are simply not reported in the published literature. And previous work within oncology, within cancer trials, has shown that time to event statistics, or survival statistics, so these statistics needed for outcomes such as overall survival, progression-free survival, and so on, are actually very poorly and inconsistently reported in the published literature. That actually hazard ratios and their measures of precision are rarely reported. So particularly for time to event outcomes, the IPD approach is definitely considered to be the gold standard for these outcomes. So the number of IPD meta-analyses that are being done over time, going all the way back to 1987, the first one that I could find, up until August 2015, when I was doing this work, are really increasing rapidly over time, as this graph shows. And actually looking from around 2009 up to 2015, and when I finished this search, it's actually an average of 105 IPD meta-analyses per year that are being reported, which is a great increase from the 90s and the early 2000s. This is definitely a method which is being increasingly used. But actually, even though this method is becoming more popular, it's still only used in around 5% of the total meta-analysis literature. The vast majority are still summary level data meta-analyses. But why is that? Considering this is apparently the gold standard approach, and there are so many advantages to doing this, why is it still only used in a minority of meta-analyses? Well, the simple answer to this is that IPD meta-analyses are really hard work. This kind of technique requires a multidisciplinary team with statistical expertise, and it can be expensive. A lot of resources are required to put together a team with the necessary expertise to do this. This sort of work is also very time consuming. I'll be describing some of the examples that I've worked in over the next few slides. So I'll try and point out some of the key dates to indicate just exactly how long this sort of work takes. And also, very importantly, I'll be coming back to this at the end of the webinar about what to do when all of your data is not available. IPD meta-analyses are open to what I'm going to call availability bias, and I'll talk about this more later on. So it should also be noted that if you can establish that an aggregate data approach can be mathematically equivalent to an IPD approach, and you can get hold of the adequate summary level data that you need from published sources, then the resource and time savings would make using aggregate data the approach of choice rather than the gold standard approach of IPD. And what do I mean by that? So thinking about choosing whether to use IPD or aggregate data for your work, a couple of things to consider. Thinking about the question and the objective of what you're doing, and also the outcomes of interest of what you're doing. So you may be interested only in the treatment effect. How does one drug compare to another? How does an intervention compare to a placebo? Or you may be interested in more complicated things than this, such as subgroups and covariates, heterogeneity, complex data types, complex statistics, and so on. In terms of your outcomes, you'll have a good idea of what the outcomes would be for your research question. Then think about, are those outcomes likely to be reported within the published literature? So, for example, time to event statistics aren't very well reported within oncology, perhaps in other areas too. And it's also worth thinking about the definition of the outcomes that you're interested in, and how they may be reported across trials, whether they will be reported consistently, or if there is any potential for outcomes to be defined slightly differently. And just to give a very general bit of guidance here, that usually, when you're thinking about mathematical equivalence, only if you're looking at treatment effect would an aggregate data approach be equivalent to an IPD approach. And also thinking about the outcomes of interest, it's only if the outcomes data is reported and the outcomes are defined consistently that an aggregate data approach would be mathematically equivalent to an IPD approach. If you have any of the other scenarios, so if you're trying to do anything more complicated than simply estimating relative or absolute treatment effect, or if there is any chance that outcome data isn't going to be reported well or consistently, then an IPD approach is probably the more sensible approach to take. So now I'm going to speak about some of the examples that I've worked on in the Cochrane Epilepsy Group. And this is the network diagram of a large IPD network meta-analysis in epilepsy. And just to talk through this plot, here we have 10 different anti-epileptic drugs which are used in monotherapies. This is single therapy treatment. So these are the drugs that would be on offer to a patient who has recently been diagnosed with epilepsy. And you can see from this plot that the size of the circles, the blue circles, the nodes, that indicates how many studies that drug has been included in. So we can see that CBZ, VPA, PHT, LTG, they're some of the most commonly used drugs. Whereas the lines in between the drugs, this indicates where there are comparisons between those drugs, and the thickness of the line shows how many patients are included in those comparisons. So we can see we have quite a lot of patients that have been included in trials comparing CBZ and VPA, also in CBZ and LTG. And also the line is quite thick, so there's quite a lot of patients in trials comparing CBZ. If you look at the top, LEV, which is a newer drug, it hasn't yet been included in that many trials, but it's been included in some very big trials. The other benefit of doing a network meta-analysis is it allows us to estimate a treatment effect to compare drugs where they haven't been compared head-to-head in a trial. So if we look over towards the left-hand side of the plot, towards OXC and PB, there is no line between those two drugs. Those two drugs have never been compared head-to-head in a trial. However, using the rest of the information in the network, we can form what is known as an indirect comparison between those drugs to get an idea of how they would compare head-to-head without actually having to do it. So in total for these 10 drugs, we have 45 different pairwise comparisons that we can make from either direct or also from indirect evidence. And also as a related side project to this large IPD network meta-analysis, we've also conducted several pairwise Cochrane epilepsy IPD reviews of these drugs. And most of these have been related to carbamazepine. I'll flick through these and the links are here to look at. Carbamazepine is a commonly used drug in the UK. And then others have been compared to PHT, phenytoin, as this is a drug which is commonly used in the United States. So for us, for this particular example, we think about our choice between IPD and aggregate data and our rationale for using IPD in this case. So firstly, I'll come to the outcomes of interest. Our primary proposed outcome for this review is time to withdrawal of allocated treatment. And we also have several secondary outcomes, time to first seizure and time to 6 and 12-month remission of seizures. So these are all time to event outcomes. We know from the literature in oncology, in cancer, that time to event statistics aren't reported very well within the literature to allow meta-analysis. So we don't know within epilepsy, but would the summary data be there? Would epilepsy be any better than the cancer trials? We're not sure. Also looking in a bit more detail at our primary outcome and thinking around the definition of this outcome. This is actually a complex outcome, as there are several reasons why someone may withdraw from their allocated treatment. It may be because the treatment isn't working for them, they're still having seizures. They may be experiencing side effects from the treatment. There may be other reasons people can withdraw. But what we're really interested in our analysis is whether the reason for withdrawal from a treatment is treatment related or not. So for example, we would class someone withdrawing because they're still having seizures or having side effects to be related to that drug, whereas somebody who moves house, leaves the trial and is lost to follow-up would not be treatment related. But the individual studies don't necessarily have the same definition as us. Some will include all reasons for withdrawal, some will only include reasons due to withdrawals for adverse events and so on. So even if we do have summary data reported in these studies that we could extract and include in meta-analysis, should we be doing that and combining this data when the outcome definitions are consistent? That's going to introduce variability, what we call heterogeneity, into our results. So also thinking about our clinical question and the objective of this work, we're also interested in establishing what the relationship is between the treatment effect and a covariate, in this case epilepsy type. And our rationale for doing this is that guideline setters, so NICE in the UK, the National Institute of Clinical Excellence, and I believe that similar recommendations were being made by other guideline developers across the world at the time. So NICE and other guideline developers recommend different first-line treatments for newly diagnosed patients with different seizure types. So for individuals with focal seizures, which is the most common seizure type, carbamazepine and lamotrigine are the recommended first treatments, whereas for people with generalized seizures, the less common seizure type, VPA, sodium valproate, is the recommended treatment. So we're taking account of this in our pairwise meta-analyses by stratifying all of our analyses by epilepsy type. And within our network meta-analysis, we want to incorporate a treatment covariate interaction. So thinking about what this means for our objectives and our outcomes, we have an interest in treatment covariate interactions. So in other words, we want to know what the relative effect of these drugs is for people with the two different seizure types. We also have complex outcomes. We have time to event outcomes, which may not be consistently reported, and the time to event statistics that we need may not be available anyway. So we've concluded in this particular example, an IPD approach is the most feasible and the most sensible approach in this case. So we've decided that we need IPD for this analysis. So we need to go about requesting this data. So I'm going to give a brief history lesson now, thinking back to the first IPD meta-analysis, certainly the first one that I can find, published in 1987. What was the landscape of data requesting like back in those early days? And I wasn't personally involved in IPD meta-analysis at this stage, but from speaking to members of the team that I've worked with who were, IPD would often be requested via a letter. You'd have to write to a colleague or a researcher conducting a trial to ask for their data, and we pick up the phone to speak to them, have a face-to-face meeting to discuss whether data could be shared. And then often data would be provided maybe by fax, maybe in the mail. Often data would be maybe photocopies on paper and would have to be computerized. And also from the experiences of my team back in the early days, data requesting was very informal. Certainly within, for example, epilepsy, everyone knew everybody else who was conducting clinical trials in this area. So friends shared data with friends without many questions being asked about what exactly would be done with that data. Whereas coming forward in time to post-2000, when I started to get involved in this work around 2011, data requesting thankfully did move into the digital age, and now requests would generally be made by email, and data would also be sent by email or maybe on CD or disk. Thankfully, I don't have any experience of receiving data on paper. But also came with this digital age, more research teams being established all over the world, smaller research teams in developing countries where they previously hadn't been. So it was no longer a case of everyone knowing everybody, and more questions would start to be asked about what exactly do you want my data for. In the late 2000s, saw the first legal documents data sharing agreements being exchanged for secondary analyses such as IPD analyses, just to protect both the person who owns the data and the person requesting the data, to give some documentation around what data can and cannot be used for. Thinking about where we are today, the present day, and also where we're going in the future with data requesting, we are now in an era of data sharing platforms. So obviously Vivli, we also have CSDR, the Yoda project, and many more out there. So data sharing platforms have been around since 2013. And then also over the last few years, there's been a wave within the research community as a whole. An era of data transparency has now really been established, with data transparency policies from some very big names within research, such as the Institute of Medicine, European Medicines Agency, the International Committee of Medical Journal Editors, and also surveys of people who conduct trials and also of patients since around 2011, compared to earlier surveys, are now showing support for clinical trial data sharing, that this is a good thing to be doing to advance research, to advance medicine. So again, just to take a step back, a little bit of a history lesson, our team has done some work in this area before, and our team published a previous IPD network meta-analysis of epilepsy trials in 2007. And for this particular project, the team requested data from 30 trials and got data from 19 of those, so 63% of the trials data was provided for. And in terms of patients, 4,703 patient data was provided out of nearly 6,000, so around 80% of data is provided, which I think is a very good IPD retrieval rate, especially considering the dates that some of these requests were done. So this would have been a lot of these requests in the days before email, so writing letters and receiving data on paper. All that considered, I think 80% is very good. In addition, IPD was available from 2 trials that were conducted at the University of Liverpool. We do have epilepsy research that goes on here as well. And then that work was done and published in 2007. Then epilepsy is a very fast-moving field. Plans to update this work as a Cochrane review, Cochrane IPD review, with newly published evidence, and also including evidence regarding 2 newly licensed drugs started between 2010 and 2011. And at the end of 2011, a new systematic search was performed and 45 new trials were identified that would be eligible for this work, identified from journal articles and conference abstracts. So for these 45 new trials, the data requests started in January 2012. This is where I was involved. I led on these requests. So how did we go about requesting IPD from these trials? What did we do? We sent a short invite letter describing our project, and also a data request form, so simply a form to ask people conducting trials if they could tick boxes to indicate which data they could provide to us. That was sent to the corresponding authors of all journal articles, both by email and by physical mail. And in the case of no response, we contacted any other author that we could on that journal article or conference abstract that we had a traceable email address for, and this did involve using some of the personal contacts of the group. We would accept data of any format. We didn't have any specific software or package that we wanted data to be provided in, and we didn't set any specific deadline for providing data. Essentially, we wanted to make it as easy as possible for researchers, for academics who may be very busy doing other things, to share their data with us, and we would be willing to spend the time preparing the data rather than asking them to do it for us. So what happened? Well, the progress over this time period, 2012-2013, was very slow. A particular problem that we ran into was around ownership of data. And by writing to authors of journal articles who are often based within hospitals or within universities, we received quite a few responses telling us to contact the pharmaceutical industry who owned the data, as only they can provide access to the data. And at the time, by visiting pharmaceutical websites, it was very difficult to know how or who to make contact with for research requests. And actually, by the end of 2013, IPD had only been provided to us for 2 publicly funded trials. These were trials conducted in academic institutions, and this was only 3% of the total data that we'd requested from these trials. So it really wasn't looking good after 2 years of data requesting about whether we would be able to even perform this work at all. Then thinking forward to January 2014, this is quite a landmark date. This is when clinicalstudydatarequest.com CSDR was launched. And initially at the time, this was a collaboration between GlaxoSmithKline and Roche. So they were the only 2 sponsors at that time on the platform. However, throughout 2014, many other pharmaceutical companies developed their data sharing policies and also put together data transparency teams, and pages launched on pharmaceutical websites, which made it a lot more obvious to know for people like me, researchers, who you should be contacting to request data and how data can be requested. So for the 16 pharmaceutical sponsored trials we had out of those 45, we made all of our requests again. We requested 6 trials between 2014 and 2016 in CSDR, and we requested 10 trials directly from the sponsors, so from the data transparency teams or from websites. And I should note at this point that at the time that we made those requests, these sponsors were not part of any data sharing platform, but all of these sponsors now are. Also, we tried to benefit from the interest, the popularity of data sharing across the entire research community, and made all of our outstanding requests to the 27 publicly funded and government-funded trials that we hadn't yet got data for, again, to see if we could stir some interest from those who maybe hadn't replied to us previously. So we made 6 requests to CSDR over this time period, and I'm just going to now briefly summarise from a researcher point of view how one actually makes a request from these sorts of platforms. So there was a list of studies provided on the platform, listed by either sponsor ID or NCT number, for example, from clinicaltrials.gov. And these studies that are listed, IPD is available to request. If the study is not listed, or if it's not obvious to find the study, perhaps if there's no NCT number available, you can submit an inquiry to check the availability of the IPD from that study, and the feasibility of providing that data is checked by the trial sponsors. Once you've established that there is some data there that you may wish to request, you then submit a scientific research proposal and also the details of all research team members. It's important, it's essential that at least one team member must be a qualified statistician, so someone who is qualified to analyse data, and also research proposals must include a publication plan. So research proposals then go to independent review boards for approval, and I believe that they're judged on their scientific merits and the clarity of the research question that is being proposed and what the data will be used for. So modifications or clarifications if the research proposal isn't clear may be requested prior to approval. If the research proposal is approved, then a data sharing agreement is signed by both the sponsor and the team requesting data, or in our case, it had to be signed by our institution, so our university had to sign that document rather than us as individuals. And then finally, anonymized data is provided in a remote analysis platform. And there is statistical software available within that platform to perform analysis, and results of an analysis done can be exported from the platform, but in most cases the data can't be downloaded or exported itself. It has to stay in there. So what happened for us next after all of our requests had been made again? So we stopped our requests at the end of December 2016. This now marks nearly 5 years of requesting. And by this time, we had managed to retrieve IPD from 15 out of 45 trials. So that's 7 publicly funded or academic trials and 8 pharmaceutical trials, which doesn't actually sound like that many, a third of trials after 5 years of requesting. But we did manage to get data from some fairly big trials, the biggest trials. And we did get data for 5,251 out of nearly 10,000 participants. And one of those trials was provided to us in the remote analysis platform of CSDR. So adding in this new evidence that we've got to the data that the team already had from the previous work, this now left us with 12,391 patients out of nearly 18,000 eligible patients, so 69% of our total data, from 36 out of 77 eligible trials to include in this Cochrane network meta-analysis. So now we have all of this data, 12,000 patients worth of data from 36 trials published over 30 years. There was a wide variety of different formats, even some different languages that the data was written in, different methods being used to collect data, and frankly different quality of data sets being provided to us. So in order for us to be able to perform an analysis of all these different data types, we needed to develop a standardized approach to preparing the data for analysis to ensure that all of our outcomes and all of our analyses were consistent. So step one for us was to check all of the IPD that we've been provided according to any published reports, journal articles, trial registries and so on. Just some basic checks to make sure that the data that we had in front of us was the data that corresponded to that journal article. So numbers of patients, checking demographics were the same, checking that some of the basic results were the same and so on. And then any inconsistencies with published work or any other uncertainties that we had about the data, we discussed with whoever had provided that data to us. And then once no major inconsistencies existed or any issues that we had could be explained, we calculated the outcomes of interest, our time to event outcomes for this work. And these two steps were done by two statisticians independently, checking and preparing the data. So we didn't measure the time accurately to prepare each data set because we didn't feel it was a fair comparison, as we had trials in very different formats, different ages, and also we didn't feel like we should be racing each other as statisticians to prepare data. We had more interest in the data being correct and prepared accurately for our analysis rather than being compared quickly. But an observation, a personal observation, as I looked at all 36 data sets that we had, the data provided from 2014 onwards, particularly from the industry-sponsored trials, definitely required fewer checks and had very few, if any, uncertainties or inconsistencies within the data sets that we needed to discuss with the sponsors who provided the data. So this meant that it was a lot quicker for us to prepare the data, and I did perceive a general higher quality of this recent pharmaceutical data, which I think is reflective of the extra attention being given to these data sets prior to sharing, such as data transparency teams within companies looking over the data and anonymizing it prior to sharing with researchers, rather than perhaps some of the academic data sets which had sat on computers for years and hadn't been looked at before they'd been sent to us. So as I mentioned, from one of the trials of 136 participants that was provided in the CSGR remote platform, unfortunately we had to exclude this trial from our IPD analyses, from our methods, as we were not able to download the data, and the methods that we were using required all of the data to be in one place. While it is possible to upload data to CSGR and to other platforms, at the time we had data sharing agreements in place with other companies, with other research groups, requiring us to have the data that we were analysing in a secure location that was under our control. So we had named a network drive at the University of Liverpool within our data sharing agreement as being the place we would be storing the data. And if we were to then upload the data to a platform where we're not in control, we would be violating our data sharing agreement. However, this was only 1% of the overall data that was included in this work, so it's unlikely to have influenced the overall results. But we did have 7 other industry-sponsored trials that were provided to us at this time, provided directly, either by email or by CD. The IPD was provided. At the time, these sponsors were not part of any data sharing platform, but today all of those sponsors are. So now 4 of these trials would have been provided by a CSDR, 2 of them would have been via the Yoda project, and 1 of these sponsors is on Vivli. So that means 7 trials across 3 different platforms. And within these trials, there's 4,534 participants, which is 37% of our total data. So that's a much larger concern if we cannot get all of that data together somehow to be able to perform our analyses. There's a much larger amount there to potentially and selectively exclude if we cannot combine data across platforms. So we've requested all of our data, we've now prepared it for analysis, and then finally we had some results, and after 7 years since the project was first planned, we were able to publish some results in 2017 from this work. So what are those results in summary? Our results support current guidelines that carbamazepine and lamotrigine are suitable first-line treatments for people with the most common seizure type, focal seizures. And they also show that one of the newer drugs, levetiracetam, may be a good alternative. This is also a good treatment. Our results also support the guidelines that sodium valproate VPA is the most effective treatment for generalized onset seizures. This is something that has been known for years, that this drug is by far the best treatment for this seizure type. But the problem is that this drug is also associated with birth defects, both physical and neurological birth defects, if taken while a lady is pregnant. So this drug is now no longer recommended for women of childbearing potential. And our results show that lamotrigine LTG and levetiracetam LEV may be good alternatives to sodium valproate for people with generalized onset seizures who cannot take valproate. So these are very encouraging results, but we must remember that these are based on 69% of our eligible data that we requested. And what can we do about the fact that we're missing 31% of data? So firstly, when we were writing our results, maybe we should have just ignored it. Hopefully no one would notice, and we would just interpret our results as if we had got 100% of IPD. And actually, this is what 25% of IPD meta-analyses published between 1987 and 2015 do. They make absolutely no reference to the fact that they didn't get all of their data and interpret results and conclusions as if they did. What we must keep in mind is this idea of availability bias. So in other words, available data doesn't necessarily reflect the entire evidence base. So in our case, does that 69% of data that we've got accurately reflect the 100%, or is that 31% different? So what if the trials without IPD are different to those that have provided IPD in terms of their characteristics and their results? And perhaps we could have a look if there's any summary data available in those trials where we don't have IPD to help us establish this. Also think about if there is any informative reason behind why IPD may not be available. Could that be related to the results? And ultimately, are our conclusions and our results from this work biased by the fact that we don't have all of this data? So we can examine each of these. Firstly, whether the trials without IPD are different to those that have provided IPD. And we can consider the characteristics and results of the trials, so such as different demographics, how big the trials are when they were published, who they were funded by, whether they have any risks of bias, positive, negative results, and so on. And in the epilepsy work, there were 36 trials with IPD available and 41 without IPD available. And from examining these different characteristics, we see that the provision of IPD does seem to be associated with the resources of the trial. So in other words, it seems to be the large multi-center pharmaceutical trials where there may be more resources to be able to make IPD available. These seem to be the trials that are providing us with more data compared to perhaps the smaller trials conducted in single academic centres which maybe don't have the same resources. And it was also difficult for us to make any assessment about whether we were more likely to be getting positive or negative results from these trials, as the majority of these trials didn't actually report the outcomes we were interested in, and we had to calculate them ourselves from IPD. Next, we can think about the reasons why IPD were not available, and if they were informative or in any way related to the results of the study. So we didn't always get a reason quoted for every study. Sometimes we were simply told that IPD wasn't available. But if reasons were given, just to briefly summarize some of them. So over the 2 time periods, so from the team that I work with who conducted the earlier review and also the work that I've been involved in, over both of those time periods, reasons quoted to us were that data had been lost, the data had not been recorded in the first place, and also there was quite a few examples of where we were not able to make contact with anybody to be able to request that data. Then some of the more recent results that I was provided with why IPD wasn't available, where data couldn't be anonymized, maybe data sharing hadn't being permitted or wasn't permitted by ethics approval forms or patient consent forms. And there was also, from the 5 trials that we didn't get from CSDR, we were told that the costs of providing data were prohibitive, so lack of resources. So we should need to consider, are these reasons informative? Are they related to the study results? That's something to think about. We can also look at whether our results and conclusions of the work we've done are biased, and we can look at this statistically. So this is a comparison adjusted network funnel plot, which is an extension of a standard funnel plot that can be used to examine publication bias in meta-analysis. This is extended to network meta-analysis. And we can use this to explore availability bias both visually and statistically. So we're looking for any asymmetry here in the data points, whether there seems to be any obvious gaps. And also we have a dotted line along here and a p-value. And this is a test of the gradient of this line, testing whether the gradient of this line is different from 0. And a significant p-value here would suggest asymmetry, would suggest publication bias, or in this case, availability bias. And so from looking at this plot, there doesn't seem to be any obvious gaps missing, no obvious asymmetry. The p-value here is not significant, so not less than 0.05. So I think we can conclude here that there is no obvious evidence of any availability or publication bias, which is good. That's encouraging for our results. Finally, we can think about whether any aggregate level data or summary level data is available for trials without IPD, as there are methods out there for combining IPD and aggregate data within meta-analysis. So looking at our epilepsy trials, the majority of them didn't report the outcomes themselves, so time to treatment withdrawal, time to first seizure, time to seizure remission at all, and very few of them reported the time to event statistics that we needed. There was some summary level data available in 10 trials without IPD, so 10 of those 41, but we did still have concerns over the definitions for time to treatment, time to withdrawal of allocated treatment, as I mentioned earlier on, about the way that different groups were defining this outcome. And we did treat that one trial that we had in the remote analysis platform of CSDR as aggregate data and combine that with our IPD using the methods described above. Actually, there was very little aggregate data available by epilepsy type, and given our clinical question to look at treatment effect across the 2 different seizure types, in fact, we were only able to add in another 2% of data for people with focal seizures and another 0.7% of data for people with generalized seizures. So this is very small amounts that we were able to add. And not surprisingly, this small addition had a negligible impact on our primary analysis and on our conclusions. So to come back to the questions that I posed a few slides ago about availability bias and how you can assess that, for our particular example, were the trials that provided IPD different to those that didn't? We found that the provision of IPD may be related to the resources of the trial, but it's difficult to know whether the results were different. And again, relating to the reasons, are they informative or not? Probably not. We don't think that data not being able to be anonymized or ethics forms not allowing data sharing to be particularly informative. But again, there does seem to be some indication here that the provision of IPD may be related to the trial. We didn't have any evidence of publication or availability bias from looking at funnel plots, and we did attempt to perform a combined IPD and aggregate data meta-analysis, but as there was a very limited amount of aggregate data available which addressed our question, this didn't change our conclusions at all. So overall, we judged that this missing 31% of data has probably not impacted on our results, and we are concluding this after all these checks that we've done. But it's very important that our results are interpreted with availability bias in mind, noting that this is only 69% of the total data and not 100%. So to summarise what I've spoken about today, I do hope this has been a useful webinar introducing IPD meta-analysis, as this is a technique which is being used more and more, mainly because the advantages are just never ending using IPD. It allows some very complicated questions to be answered, some very flexible analyses to be done, and in many cases, such as in our case within epilepsy, this truly was the only option, the only approach we could take to answering this question. It simply would not have been possible with summary level data. We should always remember that IPD meta-analysis is really hard work, and we shouldn't enter into this lightly. Requesting and preparing data requires a huge amount of expertise and is very time consuming. This one project took us 7 years to do. And the interpretation of the results from an IPD meta-analysis is directly dependent on the data that is available. Availability bias should always be kept in mind, and any checks, any additional things that can be done to explore this sort of bias always should be done. So this type of work does require expertise. While it is considered to be gold standard, these types of analyses are not automatically free from bias. And there is a new PRISMA IPD checklist, which is an extension of the PRISMA checklist for reporting of meta-analysis. And hopefully, as this checklist becomes used more and more, this will greatly improve the conduct and reporting of IPD meta-analyses. And the final thought for the day, when thinking about whether you want to embark on doing an IPD meta-analysis yourself, is to formulate your question first and think about whether potentially an aggregate data meta-analysis could answer that question, as this will save a lot of resources and a lot of time. So the note that I will end on is to IPD or to not IPD. That is the very first question that you should always think of in your research. Thank you.
Thank you so much, Sarah, for such a clear and informative lecture. I certainly learned a lot.
Great, thank you.
So I did have a few questions for you.
Sure.
And and it was very informative for me as someone who doesn't do IPD analysis, but it certainly drove home the concepts of availability bias and the need for this clearly the expertise in not just the biostatistics expertise, but clearly the statistical expertise in secondary analysis. So for those who are out there listening and maybe thinking that they have a hypothesis that they want to test maybe in an area of epilepsy or their own area of interest and therapeutic area, I might ask, if I'm a clinician with my own research team, I have a statistician on my team that has experience maybe with clinical trials, so the primary analysis of clinical trials, but perhaps not even secondary analysis. And now we have what we think is a good scientific hypothesis that we want to embark on. Is there maybe additional sophistication to start beginning this work? I think IPD analysis is maybe a little bit of a more nascent field than, of course, the primary analysis of clinical trials. So is there additional knowledge that you might impart or we need to start the secondary analysis beyond the primary analysis work? Certainly, there are areas of overlap, such as looking for missing data, et cetera, and areas of commonality, but certainly there is points of difference, such as now we would need to integrate data sets that we would be receiving, and anything else for these teams to think about specifically, or references such as the Cochrane protocol that they should be consulting prior to starting of this work?
Yes, that's a really good question. So I think the main big difference from if you are a team that has experience of collecting data for clinical trials, so this sort of primary analysis, this idea of primary versus secondary analysis, is that the team will be a lot more in control in the primary analysis of the data that they're collecting. So the statistician will probably be very key in deciding which data is being collected, the format that data is being collected in, and so on. Whereas when you're doing a secondary analysis, you still have to rely somewhat on on not knowing what the data is going to look like while you are, for example, requesting it. So if you're putting together a research proposal which is based on IPD that you haven't yet seen, you don't necessarily know what that data is going to look like, and if it is in the format that you need it to be in, or if you are able to perform any different calculations, data cleaning, analysis, and so on that can get it into the format that you need it to be in. So these are the main things to take into account. Some thought about if you're a team that is very experienced within a clinical area, then you will probably have a good idea of how data may have been collected, the structure, the format that it may be in, but you won't know for sure until you actually get it. So any research proposal that you're putting together will need to have in mind and reflect any uncertainties, and maybe the research proposal may need to be slightly changed at a later date, because maybe the data isn't exactly how you expected it to be. And as long as all of this can be documented, these are things which commonly happen in secondary analyses, that the Cochrane reviews have sections for differences between your protocol and your final analysis, because sometimes that's just the way that analysis and data is. You can't predict everything, but you should try and inform yourself as well as possible of exactly what your question is, what you're going to look at, how you're going to look at it, how that information and data may be structured, and have a plan B or a plan C if you don't end up getting exactly what it is that you thought you might get. You may need to change the approach that you were taking.
I see. So of course you have to pre-specify what you're going to do, but once you actually have the data, that may no longer be applicable, is what you're saying, right?
Yes, but I think there's pre-specification, and there is changing the methods that you're using, based on justified reasoning, maybe, for example, because data haven't been collected exactly as you thought. I wouldn't class that as being the same as completely changing your approach depending on how the data looks. So maybe you fit your model in a slightly different way, but if you're proposing a completely different research question once you've got hold of the data, then that's something else entirely.
Right, then you would have to revise. Then that would be crossing the line of that's a whole different proposal, right? You can't change a hypothesis once it's already pre-specified in the statistical analysis plan. So one of the methods that you use to search for the trials of interest that fit your hypothesis, I think that's part of your methodology. Is that correct? I mean, that seems to be what you had implied with your slides.
Yes. So certainly within the Cochrane collaboration, or doing any kind of IPD meta-analysis which involves a systematic review as part of it, so we are trying to find all of the relevant evidence, it is so helpful that we have access to information specialists, so people who are employed by Cochrane, employed by universities, who are experts in these areas. So I must admit, I don't actually do the searching myself. We have, as part of the epilepsy group, an absolutely brilliant guy called Graham, who does all of that searching for us. He's an expert in electronic databases, puts together search terms, and then sends me and the rest of the team the results to look at, and then we go through the screening process of deciding, based on what is being found on these electronic databases, which of these particular studies, particular references meet the inclusion criteria for the work that we're doing.
I see. Okay. So and it's based on the PICO terms. Is that correct?
Yes, absolutely. So it's go on.
Right. And then given that best practice dictates that the analyses need to be pre-specified, how do you go about determining what might be the best statistical methods to use, given that you don't have the data in front of you? I mean, I know you have expertise, but how would you do that?
So the methods that would be used should be based on the question, the outcomes, and any pre-knowledge that you have of what that data could look like. So, for example, in our case, we knew that we wanted to consider different different epilepsy types. So we knew that we were going to want to build in this idea of these subgroups. So that was specified in our analysis plan. And also we knew what the important outcomes were in this field, and we know what type of data that you collect to get those outcomes. So all of this informs the actual statistical approach. And because we had a very experienced team in the area of both conducting primary trials, collecting data in that setting, and also doing secondary analyses, these sort of IPD analyses previously, we had a very good idea of how the data that we were requesting would look. But we did know that we could receive data that looked like anything. There could be any format that we may receive. So, and before we actually prepared this data for analysis, we did come up with a plan, which had to be adaptable to different types of data. But this sort of plan that we had of how we were going to look over the data, how we would prepare it and calculate our outcomes to ensure some consistency, so that we weren't introducing any biases or any problems into the work from the way that we were handling the data. And this worked for us by making sure that we planned everything as much as we could in advance, rather than waiting until we had the data and then seeing what we could do.
Absolutely. Well, that certainly makes sense. Well, thank you so much, Sarah. That was so informative, and I really want to thank you for your time today.
Thank you so much. It's been great.
Thank you. Have a good day.
Okay, thank you. Goodbye.
Thank you.